So hello everyone, my name is Lucas. I am a postdoc in Brazil. I work with Noa Vazeux-Blumental at Universidade de São Paulo. And today I'm going to present to you some of the things we have been doing over the last 2 years. So, I would like to start our presentation with this famous image here from Sepkowski, where it shows marine diversity changing through time and being punctuated by the 5 big mass extinction events. And we all know that one of the main goals in evolutionary biology is try to understand the mechanisms that generate such patterns through time. And we can do that looking into speciation and extinction rates, because usually the mechanisms that control diversity, they leave some kind of signature that you can trace, look into those rates that always follow diversity patterns. And we know that these mechanisms are actually biotical or abiotic factors. And for a long time, we had this common view, an antagonized view of the red queen and the coarse jasper, that at shorter temporal and geographic scales, biotic interactions seems to be more important to explain diverse dynamics. But as we go to larger temporal and geographic scales, abiotic events become more relevant. However, over the last few years, some papers had shown us, like this one here from Peters et al. 2016, that competitive interactions can actually scale up and become important to explain diversity at longer periods of time and also at continental scales. However, only a few clades allows us to go to the species level to understand if different lineages are more or less prone to being influenced by competition during the revolution. And so this is still an open question for a lot of clades. And luckily for us, the order Carnivora is a very good group to study because they are not only cute, but there is a great fossil record of many of these lineages that allows us to ask these kind of questions and then understand better their competition and how it shaped their evolution. Besides, we have seen that biotic diverse dependence seems to be happening within families of Carnivora and also between families. This is very important for us because it shows that biotic interactions are key elements controlling their diversification, either controlling speciation rates or extinction rates or both together. But how can we actually measure competitive interactions within clades? How do we know, for example, if, take felines, for example, if competition was very strong during the evolution of phalange and was the most important factor to explain the evolution, or was actually very weak? And the most important factors to understand felines were actually competition with other families, like canids, or environmental factors. In the past, we have tried to understand the influence of competitive interactions on diversification fairly indirectly. We usually assume that as diversity increases, at some point competition will also increase as resources are limited. And at this moment, we expect to see a drop in speciation rates or an increase in extinction rates, or both things together, resulting in a loss of diversity. However, this is actually diverse dependence and not competition per se. So diverse dependence can lead us to different ways based on how we can interpret our data. So one of the main goals in our study here is try to bring to the table a more direct way to measure competitive interactions within lineages, and then apply this framework to different families of the Carnivora order to understand how competition can actually shape their diversification. And to do that, we use the morphospace. So let's take now an example with canids and a morphospace with 2 different axes. But the framework allows you to have 1, 2, or 3 different axes based on how many traits you have. So here is a morphospace with 2 different axes, body mass on the x-axis and diet on the y-axis. And diet's based on the shape of the carnassial teeth. So this value will indicate if you are more likely to be an hippo, meso, or hypercarnivorous species. So you place all canids into the morphospace, and then you can measure the distance between them using the nearest neighborhood distance. So now we know for each one of the focal species, which is the closest canid to it, and then more likely to compete with each other. But of course, we have to take into account the spatial overlap into these models, because to be able to compete, species, lineages have to be in the same place. So to do that, we have two different geographical scenarios. We have the regional scenario, that's a more permissive one, where you allow all species that live in North America or Eurasia to coexist and be able to compete. And we have the side scenario, that's a more restrictive one, where we won't allow species to compete if they are found in the same sites. And of course, species to compete, the lineages to compete, they don't have only to be in the same place, but also at the same time. So we take temporal overlap into account, looking into the lifespan of the lineages. So the way we see so far is that this framework allows you to understand how lineages perceive each other in space and time based on their ecological similarities. And well, I'll not go into all the details of the method, but you can know a little bit more about it on this preprint that Rodolfo, Thiago, and I recently published in bioRxiv. And of course, I'm open to talk about it later. But for our study here, we have 5 different time series that we generated with this framework, and we used to correlate with speciation and extinction rates. We have our regional and site measurements of diversity through time. We have our regional and site measurements of competition. And we also have an environmental factor, that's the temperature. We performed the analysis in many different ways. But here are the main 4 ways we did it. We grouped together all families that are part of the Caniformia suborder and performed analysis for all these species together. We did the same for all families that are part of Feliformia suborder. And we also did the same for all families living in North America and also in Eurasia. And just a disclaimer here, we didn't use fossil records from South America and Africa because, as many of you know, fossil records there are not so good, so at this point we didn't use so far. But besides doing that 4 types of analysis, we also perform the models within each one of the 13 different families that we used in our study, because we wanted to understand what is happening within each one of the families, how diversity is being influenced by these different time series. So we have extant species, families like canids, felines, bears, but also extinct ones like Ophionidae, Barbrophilidae, and Inhavidae. And now the fossil records, we downloaded them from PBDB, the Paleobiology database, and the Now database, and we merged both datasets and took care to exclude from the datasets pseudoreplicates that we found. And the framework was developed in R to generate the 5 time series that we used, but later the correlation of the 5 time series with speciation and extinction rates were performed in the Python software using multiple birth-death models. And now I'm gonna present to you some of our results, and they are very preliminary results and not a little bit of discussion, but keep in mind that we just got the results before coming to this conference, so they are very new to us, and there are still a lot of things that we need to figure out. But I want to show to you guys anyways. So this is the result for all the North American families that we use. And we performed analysis in 2 different ways. We did analysis for the whole lifespan of the lineages from 40 million years ago until the present. But we also did that in 4 different time windows because we wanted to understand if at different moments of time, this time series would influence the diversification rates in different ways. So we have here the 4 time intervals that are the same ones here. And they were defined by the way fossil records are distributed over the whole lifespan of the lineages. So how to read these results here? So on the upper part, you have the correlation of the 5 time series with speciation rates. And on the bottom, you have the correlation of the time series with extinction rates. Gray bars are correlations that are not significant, and colored bars are correlations that are significant. And you can know if it's a positive or negative correlation based on the orientation of the bar, related to the x-axis here. So we found some results that we would expect to find, with what we know. For example, like the green arrows indicate stuff that we would expect to find. So here we have, in 3 of the 4 time windows, we had a negative correlation of speciation rates with site diversity, which means that as diversity increases, speciation rates decrease, which is a typical diverse dependence scenario at the site location. We also found a positive correlation of our measurement of competition on the site scale, which means that as species become more different to each other, speciation rates increase. So as species become more different, less competition, and then more space to new species to be originated. And we also found a negative correlation of extinction rates with our measurement of competition on the sites, which mean that as species become more different to each other, competition decreases, and extinction rates also decrease. But we, as I said, found some results that we were not expecting, and we still need to figure out what they mean. So for example, here, we have a negative correlation of speciation rates with our measurement of competition on the site locations. That means that when you have more difference between species through time, your speciation rates will decrease. But we can look into that from another angle, and we will find the highest values of speciation rates at the moments that competition should be stronger and species are more similar to each other. So at this moment here, competition is actually allowing more species to be originated than blocking it. And we also found some of the results that we don't understand so far. For example, here we have our measurement of competition on regional scale being the most important factor to explain extinction on the last time window, having a positive correlation with extinction rates. And this is the result for the Eurasian families. And again, some of the results that we would expect to find, correlations with our measurement of competition here and here on the extinction rates, both of them. And also another results that we still need to figure out. But the take-home message here that I want you guys to take is that breaking analysis into time windows seems to be very important, because now we can see how these different variables, they influence lineages differently through time. So at some points we have more importance of temperature, the other times we have diverse dependence happening on different geographical scales. But in other moments, we have competition acting, either controlling speciation rates or extinction rates, or both together. So competition seems to be crucial in some points of time for some of the lineages. And it's really cool results that we got competition acting in opposite ways. So at some moments, competition can actually hinder diversification, as we would expect, decreasing speciation rates and increasing extinction rates. But at other moments can actually foster diversification, allowing more species to be originated. And just to remind you, these are results just for North America and Eurasian lineages. We have a lot of stuff from Coniforma and Filiforma and each one of the families. So a lot of stuff that I could not present to you guys today, but hopefully the next time. And that's it. I'd like to thank Rodolfo and Thiago for this amazing work we are doing together, and all people from our lab, and also FAPESP for the financial support and the opportunity to be here. And thank you guys so much for coming today.
Okay, so hello everyone. I'm Noa Vazeux-Blumental. I'm a PhD student at the University Paris-Saclay in the Department of Quantitative Genetics. I'm really happy to share part of my work with you today. So in my PhD, I try to understand the phenotypic and genetic basis of co-adaptation in crop mixtures, also known as intercropping. And today I will present you the result from our maize bean intercropping experiment. So to give you a little bit of context on this particular association, maize and bean have been associated as part of a traditional agro-food system known as milpa. So in milpa, we grow together the common bean that will climb on the maize, and the two are also associated with the squash. So it seems that milpa was practiced not so long after, and perhaps before the domestication of the three crops in Mexico. And there is a lot of mythologies and beliefs that were built on milpa. So perhaps you heard about the system of the three sisters, where each crop is personified into one sister, and the three grow well only when they are together. So milpa spread out of the American continent and is still practiced nowadays. And so if we try to understand what explains the success of milpa, it has been shown that several beneficial mechanisms occur among the three crops. So the first is the complementarity of the three plant architectures that will lead to a decrease of competition among the three, but also to increase the competitiveness of the system toward weeds. It also limits the spread of diseases due to physical barrier effects that will play each plant for the others. There is other mechanisms that are known as direct facilitation. So each plant modifies locally its environment, and it can happen that this modification will benefit to the partner crops. So, for instance, in milpa, it has been shown that the large leaves of the squash will maintain soil humidity. And finally, there is also what is called indirect facilitation. So each plant is accompanied with soil microorganisms, and these microorganisms can also benefit to the partner crops. So in the case of milpa, the common bean is able to form a symbiotic interaction with soil rhizobia that will form root nodules and make the bean able to fix atmospheric nitrogen that will benefit to the system. So altogether, these mechanisms can be used to design crop mixtures that will base their effectiveness on this positive plant-plant interaction rather than relying on chemical inputs. So in my PhD, I'm focused on maize-bean association, and especially on the bean response to the maize as a biotic environment. Okay, so with this presentation, I will try to answer the three questions here. So first, what is the bean response to different maize environments? Can we identify key traits in the two species that will make a efficient system? And finally, do varieties that are traditionally used in association display particular features for co-adaptation? Okay, so we set up a multi-field experiment. So the first step was to design a panel of diversity of the common bean. So I relied on genomic data of a very large panel for which I described the genetic diversity. So here you can see two main groups, and for the experiment, I was focused only in one of the two groups to avoid strong genetic structures that would have been confounding factors for the analysis. So at the end, I selected 200 European lines from the Mesoamerican gene pool, and I included 21 varieties that are traditionally used in association. The 200 bean lines were associated with 3 different maize populations that are used for human consumption. So the first one is the maize Valini. It's a Romanian variety that is also used traditionally in co-culture. There is one from Italy, the maize Spinato di Gondino, and one from France, the maize Grand Roux Basque. So we applied this design that is composed of 2 main blocks. Each block is divided in 3 sub-blocks that correspond to our 3 maize populations. And for each sub-block, the corresponding maize was sown with the 200 bean lines in randomized plots. And so we applied this design in France and in Romania for 2 years. So you have here a nice picture from the Romanian field, and we measured the traits in both crops related to phenology, yield, and development. Okay, so once we collected all this data, I started by applying some mixed model to correct for spatial heterogeneity, but I will not enter into much detail here. And so the first thing we wanted to see is that if our 3 maize population indeed correspond to 3 distinct biotic environments for the beans. So here is a multivariate analysis based on the data for the maize, and you can see 3 groups that indeed correspond to our 3 maize populations. So we have here 3 different environments for the beans. And if we look into much detail, here I try to sum up all the significant results I obtained. And so the 3 populations were different in terms of earliness. So the Grand Roux Basque was the earliest, while the Gandino the latest. They were also different in terms of competitiveness. So the maize Grand Roux Basque was the smallest, with the smaller yield that you can see below. And the Gandino corn was the highest competitive. It was the tallest, with the largest leaves, and also displaying the highest yield. And so the maize Valini, the one that is traditionally used in co-culture, was kind of intermediate. And so now if we look at the bean data, so here I represent depending on the associated maize. The first thing we see is that the beans that were associated to the Grand Roux Basque were actually the latest. So it was with this maize that we obtained the bigger offset in terms of earliness. It was also with this maize that the beans displayed the higher vegetative development and the better yield, and with the Gandino that the yield was the lowest. So here we obtained better results with the maize associated with the least competitive maize instead of the maize that is traditionally used in co-culture. Okay, so here I try to confront maize and bean yields, separating the 3 maize groups. So in the x-axis you have the maize yield, while in the y-axis you have the bean yields. And so in each cases, we obtain a negative correlation, meaning that the first interaction that we obtained between the 2 crops was competition that led to yield reduction. But the highest correlation we obtained was with Grand Roux Basque, so corroborating our previous results. And now if we look at the bean varieties that allowed the highest maize and bean yields, so they are the red dots that are in the upper right part of each plot. And so what we can see is that they were all very maize specific, and they were not necessarily the ones that are also traditionally used in co-culture. So all these varieties were just the more productive. Okay, and the last result I wanted to show you is that all these pictures of maize lodging were obtained with the Grand Roux Basque, so it was the least competitive, and so several lodging occur with this maize. But beside this, it's also with this maize that we obtained the highest cumulated maize and bean yields. Okay, so to sum up the results we obtained, we saw that the 3 maize population really are really 3 distinct biotic environments for the growth of the beans. There are few traits that were important, so we obtained the best result with the earliest maize, for which we had the biggest offset in terms of earliness. They were the lowest competitive, and yes, I think that's it. And so the best varieties were not necessarily the ones that are used traditionally in co-culture, so we couldn't have co-adaptation, but the competitiveness was really the key trait in our studies. So now we are collecting a few more data related to seed quality and also to seed morphology. And I am using the genomic data that are already used to map the success we obtain with the phenotypic data on the bean genome to identify loci that are key for maize bean intercropping. So I wanted finally to thank all the people that were involved in this work. So my 2 supervisors, Maud Tenaillon and Domenica Manicacci, and the team from the INRAE and also the University of Bacău in Romania, where we did all the field experiments. the team from Italy that provide all the bean seeds and the genomic data, and also all the fundings for this work. Thank you very much, and I will be happy to take some questions if you have.
Thank you all for coming. I'm Jenny Gleason, and as a PI, usually when we say we mean the lab. I had the fortune of having a sabbatical, and I went to Ellie Bass Lab, so when I say I mean we did this. This is all work that I did with Ellie. So usually I work on courtship behavior in Drosophila. So this was a bit of a change, starting to look at aggression levels between females. And the reason we got into this is just for what Tracy Chapman argued for earlier today, was from looking at your species. Because we were doing some experiments with a particular species of Drosophila where males were limited, and my student came into my office one day and said, the females are fighting. And I'm like, what are you talking about? And so we looked into it and found out that female Drosophila do fight, and Ellie is an expert on this, so I've been very fortunate in having the opportunity to learn a lot about this from Ellie. So, why do organisms fight? Well, the major reason for fighting is aggression. It allows access to resources. And some of the major resources that animals will fight over are mates, food, and breeding sites. And today I'm going to focus on food because it's the model that has been used quite a bit for Drosophila melanogaster and understanding how competition between females works. But there hadn't been a whole lot of work done in other species of Drosophila, particularly in female fighting. So we set out to look at the variation in intersexual aggression across species, thinking that it might be driven by resource availability. And although we didn't directly measure resources, we were aiming to test, is there variation in aggression? So a little bit about Drosophila melanogaster female aggression. There's two major fighting behaviors. There's headbutts and fencing, and I will show you what these look like if my videos work. And one of the most important things that Ellie has demonstrated is that mated female Drosophila melanogaster fight more than unmated females. And this is influenced by the transfer of sex peptide from the male to the female during mating, and also sperm itself. So both of those have a bit of an effect. So what happens in other species? We had several hypotheses going into this. One is that variation in ecology, therefore resource requirements, means that they should vary at aggression. We were expecting that post-mating change would be found in all species because it's been hypothesized in melanogaster that because once they've mated, females are going to start producing eggs and they need more food to be able to produce those eggs. So we were expecting to see that in other species as well. We also thought that aggression might be associated with reproductive traits, and that reproductive demands would similarly affect how females fight. Okay, so we video recorded mated and unmated females simultaneously, so we had those in the same group. We did one species at a time. For mating, we did the matings 24 hours before we did the contests, and before each contest, we starved them for 2 hours. This is generally because we're fighting over yeast. We want to make them so that they value that yeast. After the video recording, we track the videos using the Caltech fly tracker. This allows us to determine the location of the flies and measure per frame characteristics. From there, we put it into Java, which allows us to use supervised machine learning to score the behaviors. And we use classifiers that have been developed for Drosophila melanogaster for both headbutting and fencing. I'm only going to present the results from headbutting. Fencing is about the same. It's a lot less intense, so the headbutting is clear. And for all our behavior modification measurements, we did ground truthing to make sure that those classifiers worked, and we saw that they worked very well. And so we can take them from one species and apply them to another species. And then all the statistical analyses I'm going to show you have phylogenetic correction as well. Okay, so this is what it looks like. We are able to do 20 pairs of females at a time. And is this not going to play? I checked it, and it worked. And now it doesn't. So, one more time. Nope. All right. There we go. So anyway, that's what it looks like. And you would see that they get tracked. Okay. Oh, now it's going. Did it go? Yep. So it's usually not quite as jerky as this, but this is because it's all of them all at once, and you can see that they're being tracked. Okay. So of the species we looked at, Drosophila pseudoobscura was the most aggressive, and it's more aggressive than Drosophila melanogaster. And again, okay, so there, you see some headbutts. And a little bit of fencing, a little bit of movement with the legs. They're just reaching out and kind of touching each other. But both of those score well using the melanogaster classifiers. So we could, a little bit more. As it turns out, most species do not change aggression after mating, and those that did, which were Pseudoobscura and Bipectinata, decreased aggression after mating. Overall, most of these species, and in each case you see the unmated in blue and the mated in the red color, most of the species were pretty low in aggression. So you also see there's variation among the species in the amount of aggression, but when we do see a change, it's usually less aggression. And there's a couple that look like they're less aggressive, but it wasn't a statistically significant change. In no case did we really see an increase in aggression like you see in melanogaster. Okay, so we looked at a couple other traits. One was fecundity, and we measured that by looking at ovarials. And for those of you not familiar with insect ovaries, they are composed of these chains in which the eggs develop. And each ovarial is capable in Drosophila melanogaster of producing 2 eggs a day. So the number of ovarials in a species is really a measure of how fecund the females are. And in the species we looked at, they range from 23 to 49 ovarials. So that's between both, the sum of both ovaries. The other thing we looked at was sperm size, and sperm size varies greatly. We thought that because sperm was one thing that's transferred and affects aggression in Drosophila melanogaster, that might be a trait that might have an effect. And so these are just scaled to show you the variation in sperm size from very tiny in Pseudoobscura to huge in Drosophila hydei that we looked at. All right, so when we take these and look at aggression, we find that the number of ovarials predicts aggression more strongly for the unmated females than the mated females, but both show that as the number of ovarials increase, the number of headbutts observed also increases. Sperm size is only correlated with mated female aggression, and this is a lot less robust than the ovarials, and it makes sense that it'd only be the mated because it's after sperm transfer that there would be an effect, but in this case, as sperm size increases, aggression decreases. All right. We looked at several other traits, locomotion, lifespan, and remating rate, and didn't find any significant association between these traits and aggression. Locomotion was really important to check, so it's just not, the amount of variation that we're seeing among these species is not because they move differently. It is aggression itself. Okay. Again, it's important that you look at your organism because Drosophila nebulosa had yet another behavior, and we developed a classifier for it, and I'll show it. And this is something we've observed just holding up cultures of nebulosa. You see the males and females will do this wing flick thing, but how do I get this to go? There we go. So you see they're moving their wings, and we saw that they tended to do that more when they were facing each other and when they were near each other. And if I run the other one, and this one shows how the tracking works and that you can actually track the movement of the wings. All right, so looking at that, what we found was that the unmated females wing flick together. So if one was wing flicking, the other was wing flicking. So it might be a signal of some sort between the females, but we need to investigate this a lot more. But they do seem to be doing this simultaneously. And again, we have the mated in pink and the unmated in blue. So in summary, because I forgot to put in the conclusion slide. Melanogaster's weird, but a lot of you who work on Drosophila species know that Melanogaster's weird. It's doing something very different from the other species, and we need to be studying the other species a lot more for what the effects of mating are on them. There's variation clearly in aggression, and that aggression may decrease. Where we're going with this in my lab is now we're going to go back to that species where we think the females were fighting over the males and look at that and see if we can measure amount of aggression in association with the quality of the male. And all that remains is to acknowledge a couple people that helped us on a big day in Oxford when we were mating tons and tons of flies, and Connor in my lab filled in some of the variable numbers that we didn't have. The rest came from the literature. So thank you.